Welcome to this month's Sawaal-Jawaab session. Today we have three scientists with us who would be discussing this year's Nobel Prizes. Before we begin, you're requested to type your questions on the live chat box on YouTube. We will be taking questions after each talk. Coming to today's series of talks, the first talk is by Krishnan Harshan. He is a principal scientist at CCMB and specializes in the field of molecular virology. Today, he's going to discuss how Harvey J. Alter, Michael Houghton, and Charles M. Rice discovered the hepatitis C virus. He's also going to tell us about the impact of their work on public health. So with that, I'd like to request Krishnan to begin his talk.
Thanks, Anishila. Thanks for the wonderful opportunity. It's also a privilege for me to be speaking here, addressing a large audience on the impact of studies by three scientists that kind of revolutionized the field of human health over the last 30 years or so. Okay, so I'm a principal scientist at CCMB, and I've been working on molecular virology for several years now. I have a group at CCMB that works on hepatitis C virus, in addition to other viruses such as dengue and corona. Okay, so hepatitis C virus has been a major part of our research, and I will take you through some of the aspects of the hepatitis C virus research that these three scientists were able to contribute and then change our health. So I'm going to share the slide now. I hope it's visible to all of you. Okay. So this was truly taming a deadly viral pathogen that the story is about. And these three people, these three scientists with completely different backgrounds, they contributed to this at multiple stages of making this the research a successful one, and started with Harvey J. Alter, and then subsequently contributed significantly by Michael Houghton with molecular biology tools, and then subsequently the last phase of this entire journey was completed by Charles Rice. So they were essentially awarded Nobel Prize in Physiology or Medicine in 2020 for their contribution to efforts in finding out the root cause for a major chronic hepatitis and developing reagents that led to the discovery of the cure. Right? Now, if you look at historically, several viruses, discovery of several viruses or extensive work on several viruses have been awarded Nobel Prizes over a period of time, starting from 1954. When John Franklin, Enders, and others co-discovered poliomyelitis virus, which changed the our understanding about polio virus, and also was able to completely eliminate polio from a majority of the population. And then subsequently in 1965, Jacob and Monod, all of you guys know, they're extensively worked on bacteriophages. And Rouse actually worked and identified Rouse sarcoma virus, which was the first oncovirus to be identified. And subsequently, Delbrück, Hershey, and Luria were given Nobel Prize for identifying genetic structure of the virus. And then subsequently, David Baltimore and Taming, and of course also Delbrück, for identifying the transformation by tumor viruses. And in 1976. Hepatitis B discovery was given Nobel Prize to Baruch Blumberg, and then we know in 2008, three scientists were actually given, in fact, two scientists were given Nobel Prize for identifying HIV. So now in 2020, we have the next set of virologists who were awarded Nobel Prize for another virus, which is called hepatitis C virus. So it's an RNA virus, and it is classified under Flaviviridae family, where you would also find similar viruses such as dengue and West Nile viruses. And one thing that people do not really appreciate, that it has already infected about 170 million people worldwide and had the potential to infect a much, much larger population if there was no proper discoveries and drug development was not done. In fact, the number, if you look at it, is much more than that is contributed by HIV and MTB. So the number is actually very staggering. And essentially, the virus HCV spreads through blood or other body parts. And you can imagine that anything, blood transfusion or organ transplantation, and also injectable drug users, you would actually see that spreading, and that's exactly what happened. And it's been implicated in the development of liver fibrosis, cirrhosis, and hepatocellular carcinoma in a large majority of the infected people. And the most difficult part is that it has got 6 different genotypes. A 7th one is also proposed, but then that makes the treatment and the therapy very difficult. And currently there is no vaccine available for it. Okay? So, but the contribution by these researchers have actually led to a substantial drop in the number from about 170 million to about 70 million currently. And is still around 400,000 people die every year because of HCV-related diseases, and 1.75 million new infections are reported every year. That means even with improved drug therapy and other things, you would still find a lot of new infections happening, and that is specifically in countries where there is no surveillance or blood screening in the transfusion cases, or also in injectable drug users. Hepatitis C virus is a very small virus, which would essentially be about 50 nanometer in diameter. All it has got is a RNA genetic material inside a capsid, which is enveloped. And this is a hepatotropic. That means that it allows liver cells of the humans. It's very specific to humans as well, but it also can infect chimpanzee and some other monkeys. Now, if you look at the genome of hepatitis C virus, it's a small genome, as I said, in terms of in comparison with several other viruses. About 10 kilobase, one single-stranded RNA, which is plus stranded, and then makes one single polypeptide, which is subsequently cleaved and makes about 10 proteins. That's all. And with this repertoire of about 10 proteins, the virus is able to cause such a big problem in human beings. And the virus enters the cells through endocytosis, and it forms, it associates with cholesterol on the outside, and that is actually very important for the virus to be infectious. Now, when I come back to this viral hepatitis, as the name indicates, the meaning of hepatitis is inflammation of liver, and you can have literally 2 different types of hepatitis, which is either acute or chronic. And people have actually currently identified that there are 5 different viruses that can cause hepatitis, starting from hepatitis A to E, of which A and E have been shown to be spread mostly through water or food. And hepatitis D virus, which is Delta virus, is actually not directly causing any disease. It actually helps hepatitis B virus to make this, the symptom very severe. And then, as you see, hepatitis B and C are chronic viruses, and both of which actually spread through blood or body fluids, right? So, in terms of therapy, there's no vaccine available for hepatitis C virus because of the complexity of its diversity, as well as the ability of the virus to mutate constantly. But fortunately, with the discovery of several inhibitors, polymerase, NS5 polymerase inhibitors, NS5B polymerase inhibitors, and protease inhibitors, the advent of these inhibitors, there has been beautiful therapy, and over 95% of the cases can actually be treated. So this, when you reach at this stage where there is even though there is not a vaccine, but a very effective drugs are available to limit or prevent the replication of the virus, and then give the person a happy rest of the life. That's actually a remarkable change, and that's precisely what these people were, appreciated for. Now, I will start with how this discovery actually unfolded. So when Harvey Alter, who was actually a physician and had been working with NIH for several years, since 1960s or something, he was also involved with the identification or the discovery of hepatitis B virus. Okay, so he kind of knew in that field, he really loved liver and liver-associated things. So he had been working on several of these hepatitis, and then they consistently noticed, even after the discovery of hepatitis B virus, which happened in the late 1960s, they realized that So what changed with hepatitis B virus is that there was now a serology test with which they can actually screen the blood of the people who donate blood or even organs. So they could identify that this is a virus, and then that actually took away about 30% of the loads of the infection cases. Now, until then, this transfusion associated hepatitis was actually a big problem, and people could easily associate that to be more of a contagious than something else. So now the discovery of hepatitis B actually was a big relief. But in spite of its discovery, several cases of transfusion-associated hepatitis kind of kept on going up. Now that actually put them in a puzzle, and then Harvey had been working on it for a long time, and he really knew in and out of hepatitis B. And then by the middle of around say about 1975 or something, they actually, another group actually identified hepatitis A virus. So, but then interestingly, when they were looking at the transfusion-associated viruses, hepatitis A virus antigens could not be picked up. So that kind of ruled out the contribution of hepatitis A virus from the chronically infected or transfusion-associated cases. Now, so that in spite of screening out both hepatitis A and B cases, the transfusion-associated hepatitis was actually ongoing, and that gave rise to the thought that there is yet another unidentified factor. Even at that point of time, they had no clue that this could be a virus. So they coined this term called non-A, non-B hepatitis virus, even though they had several reasons to believe that this is infectious. Until they put chimpanzees into the studies, they couldn't prove it. So how chimpanzees use of chimpanzees revolutionized the field was, now could actually take out these blood samples from which you screen out hepatitis A and B virus cases, and then whatever is left, non-NA, NBH samples, they put into chimpanzee, they injected into chimpanzee. And very interestingly, chimpanzee developed symptoms similar to what patients found in humans, and that gave quite a lot of strength to the study and also confidence for carrying out further studies. Now, with the help of these chimpanzees, they could actually show that this is something contagious, and that is actually transmitted through the blood. Now, even though this discovery happened in around 1975, and a lot of studies actually were being carried out in chimpanzees, they couldn't actually come out, they couldn't identify that it's actually a virus. And that's primarily because there was no serology test available for this. And nobody could actually find out a factor or an antigen that was exclusively present in the infected cases, not present in the healthy persons. So that had been a major roadblock. Right. So until about 1989, there was there was progress happening, and they could actually convince the people that there's a factor non-hepatitis B. But then this pathogen was never identified until Michael Houghton, who was working in industry, Crown Corporation, for several years. So this corporation was funding several studies in the 1980s and all, and then Michael Houghton was a premier scientist in that, and he also had been working on several molecular biology tools, right? So they had several groups, and then he had been trying independently to identify certain factors or get clones that would actually express certain factors that contributes to or is responsible for this NA-NBH. Right. So primarily what they have been trying was they were trying to generate these cDNA expression libraries with the hope that at least some of these clones could actually express certain factors coming out of the pathogen or the bug responsible causing NA-NBH. But please remember this, please note that at that point of time, they had no clue that this is a virus. They had certainly some indications, but there was no evidence for it. Forget about the clue that it was also an RNA virus. So irrespective of that, they were expecting that if this is a pathogen-driven thing, they should express specific mRNAs, and that should be, if we are able to capture that, we should be able to express some, and then subsequently we could develop serology tests against them. That was a drive on which they were actually working. After several rounds of failures and multiple different kinds of approaches, finally they were able to, they hit the jackpot, right? And if you look at this, this is pretty much what they did. They used the samples from chimpanzee infected with this blood sample from patients, and they created cDNA libraries in bacteria. Remember, there was also luck in that, because they were actually using cDNA libraries in bacteria to express this factor. And fortunately, one clone, 511 that they cloned, that actually picked up a signal when antibodies, again, from the patients were tested. And that gave the first clue that, okay, that gave the hint of the first time a factor that is not associated with the host, but associated with something outside, and that could be a potential pathogen. And then they subsequently refined their technique and then finally identified multiple clones and then developed serology tests for against this. And that kind of really revolutionized the field, because here you have a system to screen blood again, right? So imagine that they took about 15 years from the original hypothesis that it is actually potentially a virus that is different from hepatitis B. And this again was a very revolutionary work. And then he and Harvey started collaborating in confirming that through samples from multiple chimpanzees and also patients, really strengthening their data, confirming that this is indeed a new pathogen that they have done. And then subsequently, they also went back and then identified that this is the pathogen has RNA as the genome, and also were able to show that this is about 10 kilobytes in length. So this study was certainly one of the most revolutionary studies after the initial discovery of the known hepatitis. Right now, so a lot of people actually started working on it, but then they were also trying to set up systems to culture the virus. Now, one of the primary things in virology is to be able to culture a virus from a patient. For example, now we have coronavirus in our laboratory, generated from the samples, nasopharyngeal samples. So we were fortunate that we already had some clue about how to do it from earlier work. And we also had the tools to do that. Right? That means we need to have right permissive cell lines to do, in which the virus should also grow. And unfortunately, this was not true for hepatitis C virus. No matter whatever you try, several people in different labs across the world tried getting the patient samples, which had tons of viruses in them. But then they would infect human hepatocytes or hepatoma cell lines. They would never get a system where the virus would actually replicate. And in the absence of a system where the virus replicates, you are never going to be able to understand the life cycle of the virus, and you are not going to be able to understand the functions of viral proteins, and you're also not going to be able to have a cell in vitro system against which potential drugs can be tried. And that's when Charles Rice, who was working at Rockefeller, actually contributed. So, like several others, and his laboratory was also trying to set up this cell culture system, and he happened to be, certainly he happened to be lucky, because he happened to also have a cell line which allowed the replication of the virus. Right? That means this particular cell line had a very low innate antiviral response, and this happened to be complete serendipity. But what he did was he was able to clone the entire HCV genome into a plasmid, and then make an infectious clone from which you could actually make viral RNA by in vitro transcription. Use that, introduce that into these cells to make more and more virus. So here, two things actually came together. The system that he developed, the plasmid-based system, and he was also lucky to have a right cell line in his hand, which happened to accommodate the virus. And also, the genome that he cloned also had certain cell culture adaptation mutant. That was another point that also happened to be lucky. Right? Now, so whatever it is, the hard work and the luck actually played a big role. And once this was actually developed only in 2005. So remember this 1975 to 1989 dry period. And from 1989, when HCV was identified, until 2005, there was no way that people could actually look at the life cycle of the virus. Now here you have a system with the help of Charlie Rice, and subsequently now several other people also reported, successful replication of HCV in cells. So these revolutionize the field again, because now you could actually study the roles of several viral proteins and could specifically target each of them. Because now you have a system, you can infect cells with the virus that are grown in the culture, come back with potential drugs and see if they inhibit the viral growth. And this actually in the next 5 to 6 years, a lot of actually development happened in academia as well as in industry, and that resulted in the development of several very, very efficient direct acting antivirals as listed here. And now currently a combination of several of these is what is used by the physicians. And in a matter of about 2-3 years, I mean, it was originally approved by FDA in 2016 or something. And you already start seeing a lot of change in the field, because I personally know several people who have now recovered from HCV, which used to be very, very extremely rare case until this was introduced. So the fate of these people was kind of guaranteed that they are going to develop either fibrosis or cirrhosis or hepatocellular carcinoma, which all of them could be fatal. Now you have a combination of drugs that are very effective against the virus. Unfortunately, I mean, that was a fortunate side of it. Unfortunately, these drugs also kind of completely killed the field of HCV research, because now you have very efficient drugs and there is no more interest in studying the biology of the virus because it's in control, right? That's on the flip side. Now, anyway, so the contribution of these people is a wonderful, wonderful story. It's like a fairy tale kind of a thing, which you don't really find in several other cases. It's about wonderful story about the discovery of an etiological agent, which was originally, nobody had a clue that this could be a virus at all. And with the help of these 3 people, now you know that it is a mysterious disease is caused by an infectious agent, and that to a virus. And then you know that you identified the virus and you understand the properties of the virus, the preferences of the virus, and now also could actually develop drugs against them, so that a lot of people were now saved. Because I talked about the 170 million, approximately about 170 million people were infected, which was about 2% of the total population at one time. Now, if that was uncontrolled, there was no discovery of hepatitis C virus. And if that went uncontrolled, a large number of almost about 1 billion people would have been infected by this, of which a majority of the population would have gone into developing liver cirrhosis or the cancer. So their contribution has been enormous, and I can't disagree with the selection by the jury. And this is no sooner also. Anyway, this discovery has been, their discoveries has been having so much profound impact on the human life. And I would like to leave with this of sentences from a perspective written by Harvey. What he writes is this: my life has been a dream. The only problem is that it has not been my dream. I never dreamed about going into research. I never dreamed about discoveries or winning prestigious awards. These wonderful things were never in mind set. It just happened. So what he's talking about is that if you could set your eyes on awards and other things, you probably don't end up being there. And that also gives a strong message to us that what we all have to do is to be inspired about what we have been doing, and then a result is going to follow us. Success is going to follow us, and I can't actually disagree with this at all. I mean, I totally agree with this, and I would like to stop here, and then I can take some questions from the audience if there are. I hope I'm not late, and thank you all.
Thank you, Krishnan, for this informative talk. In the interest of time, we'll be able to take one question now, and then we'll move on to the other talks, and then we'll ask the other questions at the end of the program. There's one question which has come through messages. It says, why was it not possible to replicate the hepatitis C virus in labs?
Yeah, that's actually a very interesting question that we all kept asking. Why don't they replicate? At least one clue that subsequently people knew are there. So when the virus enters the human body and then finds its way to liver and then enters it, it finds a very beautiful niche inside the cell. For whatever reason, we were unable to provide that through the in vitro system, in which we were culturing primarily liver cancer cells, human liver cancer cells. right? Because the primary hepatocytes don't divide, and then they are not actually a good system for infection in vitro. They have never been shown to be very successful. For whatever reasons that we do not understand, there is a big difference between in vivo and in vitro. And because of a various set of factors, nobody was able to succeed. And they required some kind of cell culture adaptation mutation that people later identified. So once you introduce these mutations, a lot of these viruses are able to replicate. But in reality, in the infected person, the sequences are quite different from what we do in the in vitro culture.
Thank you, Krishnan.
Shall I leave now?
Yeah. If we have any other questions, would it be okay if we take it at the end of the program?
I would be having to go to my interview, so.
Okay. So we'll then get in touch with you later by email if it's possible.
Yeah, sure.
Okay, sure. We will definitely make sure that all the questions, whatever we receive throughout the course of the program.
Yeah, sure. We can pass it on.
Okay. Thank you so much.
People, a wonderful talk.
Yeah, thank you. So we'll move on to the next talk of this program. So this year's Nobel Prize in Physics was shared between mathematical physicist Roger Penrose and astronomers Reinhard Genzel and Andrea Ghez for their contributions to the study of black holes. Our next speaker, Parameshwaran Ajith, who is an astrophysicist at ICTS TIFR, is going to give an introduction to the physics and astrophysics of black holes, focusing on the Nobel laureates' contributions. So without further delay, over to you, Ajith.
Thank you, Anishila. I'm very grateful for this opportunity to talk to you today. And I'm going to switch to a new topic that has almost no significance to human life. Still, black holes have captured the imagination of a lot of us in a variety of different ways. And this year's Nobel Prize was shared between Roger Penrose, who is a mathematical physicist who established some of the very rigorous proofs that black holes could exist in the universe, and two astronomers, Reinhard Genzel and Andrea Ghez, who led two teams of astronomers who made some remarkable set of observations in the last 25 years that very conclusively established that there is a very massive and compact object at the center of our galaxy that is most likely to be a supermassive black hole that is weighing millions of times the mass of the sun. So Roger Penrose's Nobel citation was for the discovery of that the black hole formation is a robust prediction of the general theory of relativity, while Genzel and Gez's citation was for the discovery of a supermassive compact object at the center of our galaxy. So as you know, black holes are among the most exotic and enigmatic objects that we know of. The existence of black holes was predicted by the Albert Einstein's general theory of relativity, which is the most accurate theory of gravity that we have right now. This is a very different picture, and Einstein's theory presents a very different picture of gravity as compared to the familiar notion of Newtonian gravity. We know that Newton describes gravity as a force that is proportional to the mass of the objects involved and is inversely proportional to the distance between these objects, etc. While Einstein's picture, gravity is a manifestation of the curvature of space and time. And any massive object, like an apple, or other forms of energy, also like electromagnetic energy, can produce a curvature on the space and time. And even more strangely, in Einstein's theory, space and time are not independent objects. They are part of a single entity called space-time. And what we call space and what we call time depends entirely on the observer, according to Einstein's theory. While this sounds all very exotic, the theory has been verified by a number of remarkable laboratory experiments as well as astronomical observations. In fact, the theory has passed all the observational tests with flying colors. Basically, Einstein's theory presents a geometrical theory of space-time, which also happens to be a theory of gravity. And this is the core of this theory is the set of equations which are written in a very compact form like this. These are called Einstein's equations. So the left part shows a quantity, a mathematical quantity, which describes the curvature of space-time. And this is called Einstein tensor. And the right hand describes the matter contained in the space-time in the form of mass, energy, momentum, etc. And this, very famously, John Wheeler, who is a famous general relativist, put this. This equation tells how, so space-time tells matter how to move, and the matter tells space-time how to curve. And both these aspects of the curvature of space-time as well as the motion of objects in that curved space-time is encoded in this very compact set of equations. Although these equations are very compactly written, in practice, this sets a set of 10 coupled nonlinear differential equations, which are notoriously hard to solve. In fact, still there are only a handful of solutions exist for these Einstein's equations. But very interestingly, within months after Einstein published this theory, another German astronomer or scientist, physicist with a polymath, Carl Schwarzschild, published the first rigorous, exact mathematical solution of the Einstein's equations. And it turns out that his Schwarzschild solution describes the curvature of space-time around a spherically symmetric object. And neither Schwarzschild nor Einstein, realized the physical importance of such an equation. They both thought of this as a mathematical curiosity. But eventually, by studying Schwarzschild's solution, mathematical relativists of the mid-20th century realized some very, very interesting things. One is that they realized that this solution encompasses a particular surface, which is called the horizon. And if the space-time encloses a particular mass, a mass which is called m, then this horizon is at a radius called the Schwarzschild radius, which is equal to 2gm by c square, where g is the Newton's constant of gravity and c is the speed of light. And this horizon contains shows a very interesting property. Things can go only inside the horizon, but nothing can come outside the horizon, including light. And it turns out that whatever things that fall into this horizon, including particles, TV sets, or electromagnetic waves, or gravitational waves, whatever, they all fall through and reaches a central point, which is called a singularity. And the singularity is a very enigmatic point. And at this point, all known laws of physics break down. We do not know how to describe singularities properly using any physical theories of the current physics. So, for example, the Schwarzschild radius, for example, that of the sun is about 3 kilometers. This means that if you are able to compress the sun into a radius of about 3 kilometers, this strange surface called this black hole horizon forms, and then not even light will be able to escape the surface and to reach the external observers. And another very interesting question that a lot of mathematical physicists in the mid-20th century asked is that, can black holes exist in the physical world? After all, the Schwarzschild solution is just a mathematical solution that describes a solution to a set of equations. It is a well-defined solution, but does it have any physical significance? Do objects described by the Schwarzschild solution exist in the physical world? Can they exist in the physical world? And this was answered by, this was a scene of a great controversy in the 1930s and 40s. And this was settled by a series of papers at first. And I'll discuss this research, but the conclusion is that it turns out that massive stars, which are several times more massive than the sun, can collapse at the end of their lives to form objects like black holes. And let me give you a quick 101 on the theory of stellar evolution. And we know that, for example, stars are in equilibrium. Their inward pull of the gravity is balanced by the pressure of the hot gas as well as the light produced by the star, the pressure of the outgoing radiation. These two are in balance and they're in an equilibrium. But at the end, once this nuclear fuel of the star exhausts, there is nothing to produce a pressure to balance the act of gravity, and the gravity dominates, and the star can collapse. And depending on the mass of the original star, the final object, the remnant object, could be a different types of compact object, like it could be a white dwarf or a neutron star or a black hole. And it turns out that, and in all these cases, in white dwarfs and neutron stars, the pull of the gravity is balanced by other kinds of pressures, which are called degeneracy pressures, which is a property of quantum mechanics. But it turns out that when the star is more massive than a certain number, about 10 times the mass of the sun, no other degeneracy pressures can support the inward pull of the gravity, and this core can only collapse into singularity, forming a black hole. This was first established by, famously attributed to a paper by Oppenheimer and Snyder in 1938, who showed that a spherically symmetric distribution of dust would, with appropriate conditions, would collapse and form a black hole. And interestingly, it turns out that another Indian physicist called B. Dutt had published a very similar mathematical calculation a year ago, but very little is known about him. His paper was sort of not recognized until very recently, except for some very obscure citations in certain books. But it turns out that this very enigmatic young person who died very soon after he published this paper also made a very similar contribution, which is famously attributed to Oppenheimer and Snyder. But the summary is that when all thermonuclear sources of energy are exhausted, a sufficiently heavy star would collapse into a black hole. But even these papers were viewed with a lot of skepticism by the leading physicists at the time. The reason was that they assumed certain very idealized conditions of things like spherical symmetry, etc. And it was not clear whether gravitational collapse or the actual stars in the physical world would obey such idealized conditions. And here comes Roger Penrose. In a paper that he published in 1964-65, he showed that if you just assume some very basic physical assumptions, one could show, using some very rigorous mathematical tools that he developed for this work, that a sufficiently massive object will collapse under its own gravity to form a singularity at the center. This is called a singularity theorem that is attributed to, that is first proved by Roger Penrose in 1965. And this basically set the mathematical theory of black holes in a very, very rigorous footing. And this was successively developed by other people like Hawking, who proved a set of very, very interesting theorems of singularities involving even the whole universe, in the Big Bang singularities, etc. Again, there is an interesting Indian connection. Roger Penrose brought in a lot of very interesting mathematical tools, topological tools to prove this theorem. And also, he used this calculation based on the so-called Rai-Chowdhury equation, which was developed by A.K. Rai-Chowdhury, an Indian physicist who lived in Kolkata and taught in the Presidency College. So there's an interesting Indian connection here as well. So people like Roger Penrose, the mathematical relativists of the mid-20th century, were trying to answer the question, can black holes exist in the physical world? But now you can ask a very different question. Do black holes exist in the physical world? And this comes in the regime of observational astronomy and experimental, etc., because this calls for empirical evidence. And starting from 1960s, a variety of astronomical observations suggested that objects like black holes must exist in the universe. And the first clue came from the observation of very bright radio sources called quasars, which seem to come from very, very distant universe, almost at the edges of the visible universe, billions of light years away. And these appear to be very, very bright, and at the same time, very, very distant. So the only conclusion that these objects have to be intrinsically extremely bright. And people tried to calculate or guess what kind of processes would power such extremely luminous objects. And it turns out that the only thing that can power this kind of luminosity is basically the gravitational potential energy released by infalling matter to black holes. This is the best explanation. No other explanation, including nuclear physics, could come up with this kind of energy budget. Just like these radio astronomers observe these extremely bright objects at the very distant universe, X-ray astronomers also found similar objects in a much micro size called micro quasars from our own galaxy also. And here again, the best explanation that these are much smaller black holes, black holes that are still 10 times or several times more massive than the sun, accreting gas from a companion star, and this accreting gas gets heat up, producing these X-rays. While these quasars are believed to be powered by supermassive black holes, which are millions of times as massive as the sun. Another very interesting arena for finding a black hole was our galactic center itself, the center of our galaxy, which is known to be in the constellation called Sagittarius in the southern hemisphere. The left plot shows a cartoon picture of our galaxy. We cannot actually take an astronomical picture of our galaxy because we are in the galaxy itself. So, and you can see our location. This is the sun is marked here, and around the sun is our solar system. But if you just astronomers looked at this galactic center, which is the middle part of this spiral galaxy, and the right plot is an actual astronomical photograph. And the center is marked by another yellow square, and this is known to astronomers for almost a century that this is the galactic center, and people have found a very bright radio source, which is called Sagittarius A star. And starting from the 1990s, a couple of teams of astronomers started measuring the motion of stars around the galactic center, and they have found something very, very remarkable. They found that many stars are moving extremely fast around the galactic center. And in particular, some of the stars completed their orbital revolution within a matter of a decade or so. So this is not an animation. It's actually an animation made by a large number of actual astronomical exposures taken by some of the best telescopes across the world. And over a period of like 25 years, this particular animation is due to the UCLA group led by Andrea Ghez. And you can see that many stars have completed one orbital revolution during a period of 16 to 20 years. And many other stars are in the process of completing their orbital revolutions around some unseen object, which is at the galactic center. And just like we can calculate the mass of the sun from the motion of planets, we can calculate the mass of the central unseen object from the motion of these stars. And using some very simple laws, like some Newtonian physics. And when these astronomers did that, it turns out that the central dark object has a mass of about 4 million solar masses, 4 million times the mass of the sun. So our galactic center has a dark secret that is extremely massive, which we cannot see. And the only explanation that astronomers have is that our galaxy hosts a supermassive black hole. So this was a true defeat in the observational astronomy because it's a very, very difficult measurement. It's because the galactic center is obscured by dust, and it's very hard to observe these stars. And these astronomers needed to go to near infrared wavelengths to observe the stars. And they developed some very, very sophisticated technology, like adaptive optics, to make the precision measurement of the location of the stars using some the most expensive and state-of-the-art telescopes in the world, like the Keck Observatory, as well as the European counterparts of the Keck Observatory, which is called, I forget the names. So anyway, the two teams of astronomers, Reinhard Genzel of the Max Planck Institute and Andrea Ghez of the UCLA, were awarded the Nobel Prize for discovering this presence of this extremely massive and compact object at our galactic center, which is very, very likely to be a black hole. I was careful in using that even the Nobel Prize committee was very careful in saying that they say it's an extremely compact and massive object at the center of the galaxy, not a black hole, because we do not, these observations do not have the kind of power to tell whether these massive and compact objects at the center of our galaxy is indeed a black hole or something that is more exotic, that is, could be as massive and compact. But recently, many other astronomical observations are starting to probe the real nature of these objects, which we believe are black holes. And the left plot shows an example of the first gravitational wave observation by the LIGO observatories, which in 2015 discovered a gravitational wave signal produced by the collision of two black holes in a very, very distant galaxy, about 1 billion light years ago away. And these observatories are still taking data. So far, they have detected dozens of such signals from different black hole collisions happening at different parts of the universe. And these gravitational wave signals are starting to probe some real detailed nature of the actual nature of these compact objects, which we believe, again, are black holes. The right side shows the image of the black hole at the center, supermassive black hole, the center of our neighboring galaxy, M87, which hosts another black hole about 1 billion times the mass of our sun. This was taken by a collaboration of radio telescopes called the Event Horizon Telescope. This was released last year. And you can see that this dark shadow among this luminous disk of matter, which is this infalling gas producing electromagnetic radiation, and while this gas is falling into this supermassive black hole. And for the first time, we are having really a shadow of the region around a black hole, a few times the Schwarzschild radius of the black hole. A similar picture of our galactic center black hole is expected from the same collaboration in a matter of a month to a couple of years. And then we have much more conclusive evidence that what we have seen, what we have at our galactic center is indeed a supermassive black hole. Let me summarize now. A quick summary is that a variety of astronomical observations suggest the existence of massive and compact objects that are consistent with being black holes. However, the real smoking gun evidence of a black hole in general relativity is the existence of singularities covered by a horizon. And we do not have any empirical evidence of either singularities or black hole horizons yet. But hopefully, in the next decade or so, we will be getting the first evidence of the physics of this, around the black hole horizons. And astronomers also know that there are about 10 million such stellar mass black hole-like objects in our own galaxy. And these are likely to be produced by the collapse of massive stars. We also know that each galaxy hosts a supermassive black hole. Most of such galaxies like ours host a supermassive black hole, each weighing a few million times, billions of times the mass of the sun. However, we do not know how nature is producing these supermassive black holes. There are some theories, there are possible explanations, but none of them are fully satisfactory yet. There are some observational hints of medium-sized black holes, so-called intermediate mass black holes, that are in between the stellar mass black holes and supermassive black holes. These intermediate mass black holes can be a few hundred times the mass of the sun. There are some very interesting theoretical predictions of what is called primordial black holes that can be extremely tiny as well. Black holes can be as small as the Earth or even smaller, and could be very giant as well. And these are likely to be produced in the very early universe because of the quantum fluctuations at these highly dense parts of in the early universe. Some very high dense parts can form, and they can collapse under their own self-gravity, producing primordial black holes in the very early universe. This is a theoretical prediction, which is yet to be verified by astronomical observations. Black holes also offer a number of puzzles for theoretical physics. For example, we do not understand singularities. We do not know how the information is lost into black holes. This is a very interesting puzzle called the black hole information paradox. There are no there are arguments explaining these ones, but there are no conclusive understanding of these puzzles yet. Perhaps we will need a quantum theory of gravity to explain these aspects of black holes. Meanwhile, while theorists figure out these issues using theoretical calculations, astronomers also developing new tools for probing the real nature of these extremely compact and massive objects that we know exist in the universe. So it's a very exciting time in the study of black holes, where a variety of theoretical, mathematical, computational, as well as observational astronomy approaches meet to study different aspects of this very, very exotic and very interesting objects. So let me stop here. Thank you very much.
Thank you, Ajith, for this really engaging talk. We have around 7-8 questions.
Okay.
But would it be okay for you if we ask one now and we visit the rest at the end of the program?
Absolutely.
Okay?
Absolutely,
okay,
yes, yeah.
Okay. So I'm just asking one question right now. What is the time scale over which the exhausted star turns into a black hole? Is it instantaneous?
No, well, the actual collapse, the stellar evolution happens over millions to billions of years. But once the stars start to collapse, this can be very, very, very short-lived. It could, the collapse can happen even timescales of sub-seconds for the case of massive stars.
Okay. So Ajith, we'll take the rest of the questions after the third talk.
Sure.
And for now, we'll move on to the third talk of the day. The next speaker is Sonal Nagarkar Jaiswal. She's a faculty at CCMB who is working in the field of neural development using Drosophila and human neuronal stem cells as a model system. Today, she's going to discuss the CRISPR-Cas9 system, a path-breaking genome editing technology developed by Jennifer A. Doudna and Emmanuel Charpentier. Now, I would like to request Sonal to please begin her talk.
Thank you, Anushila. So today I'm going to tell you how Emmanuel Charpentier and Jennifer Doudna's lab, they invented a new genome editing technology that actually came from a basic research project. The technology is called CRISPR-Cas9, and it has revolutionized the whole genome editing field. So what is genome and what is genome editing? So genome is the genetic material of an organism, and it consists of DNA or RNA. And the genome could have a few hundred to hundreds of thousands of genes in any organism. So basically, genes are long strands of DNA that are composed of nucleotides, and that they carry codes that decide which protein should be expressed. So for protein expression, so DNA undergoes 2 rounds, and the first process is called transcription, which give rise to a single strand molecule, which is called RNA. And RNA have these triplicate codes for different amino acids that upon translation form a long polypeptide chain, and which then folds 3-dimensionally and give rise to different proteins. And these different proteins are involved in many different cellular processes. So, but then so as I told you that there are hundreds of thousands of genes. So how do we know which gene codes for which protein and where is it involved? Like with the function of gene, how do we know? So the best way to find it out is basically we perturb the function of a gene and then look for cellular processes that are affected. And that's how we find out its function. So what are mutations? Mutations are basically nothing, a single base change in DNA. But here I have an example. So or deletion of a base, which actually leads to an abnormal or no protein formation, which perturbs the function of this gene. So for example, here I have this A, which was C, and now it is changed, which actually brings a premature stop codon here, and it leads to a truncated protein. So basically now we have this protein which is truncated. It's not functional, and it will disrupt the cellular function. So how do we know? So there are hundreds and thousands of genes. So how do we get these mutants for all these genes? So there are several efforts had been made to find out or to basically generate mutants for many genes. The first one started with the first mutation were basically created by a person. His name is Hermann Joseph Müller in 1926, where he used X-ray to generate mutants of small flies, fruit flies that are called Drosophila, and for which actually he got Nobel Prize in 1945. So, however, these mutations were not a single deletions. There were large deletions and many large chromosomal rearrangements. So then people turned, and they were random. So people turned to different things like chemical mutagens, where they use different mutagens like EMS, and they fed animals and then screen for mutations, random mutations. Then we also have biological agents like transposon. So transposons are jumping genes. They basically hop from one chromosome or one genomic locus to another, and thereby they create deletions or insertion mutations. And this method also got a Nobel Prize. So Barbara McClintock actually discovered jumping genes for the first time, and she got Nobel Prize in 1983. So, however, all these methods, they generate mutations, but they are random mutations. And to find out what is the precise location of mutation, we'll have to do DNA sequencing or different complementation analysis to figure out where the mutation is and which gene is affected. So, in the same line, there was one method which came out, and that is the RNA interference. So RNA interference is in RNA interference, we can actually target a single RNA molecule for a specific gene, and which will lead to degradation of this RNA, and there will be no protein produced. So which will again impair the function of this gene. So this RNAi method, it was again got a Nobel Prize. So Andrew Fire and Craig Mello, they developed it, and then they got Nobel Prize in 2006. So we can already see back to back, our desire to understand and learn how gene function that led to so many great discoveries, and it is still continuing. So in the same line, then there were new methods were developed, and these are the genome editing nucleases, meganucleases, ring finger nucleases, and talons. These were much better than the previous methods because they could actually still do very close to the desired area where we want to create mutation. However, these are protein-based methods that actually applies protein domains that actually interact to a specific DNA region. So therefore, the problem here is, though we can make desired mutation or close to desired point, we'll have to do a lot of protein engineering. First, we'll have to detect the sequences that actually we can use for protein binding. Then we'll have to engineer proteins according to those sequences, and then they will create these breaks. So while we were working on these, using these methods to create mutations for different genes, there were 4 groups. They were actually working, or they were developing a very cool technology, which is called CRISPR-Cas9, basically a tool for rewriting the code of life. So these 4 groups were in 4 different places: Jennifer Doudna's group, Emmanuel Charpentier's group, Virginius Sixnick's group, and Feng Zhen's group. So all these groups were independently working on the system, which is called CRISPR-Cas9 system, that got the Nobel Prize this year. So for that, Jennifer and Emmanuel got the Nobel Prize this year. So what is this CRISPR-Cas9? The CRISPR-Cas9, so let me tell you what it is first, and then we'll see how they used it. So the CRISPR-Cas9 system is basically was found or is found in bacteria or archaea. It has 3 major components. The first component is the CRISPR loci, which you can see here. This CRISPR loci has several palindromic repeats. They are present in a cluster, and they are spaced or separated by small random spacer sequences. And then there is a leader sequence that is present just before these locus. This leader sequence contains a promoter that actually helps in transcription of an expression of this CRISPR loci. Then second component that we have is Cas9 gene cluster. So Cas9 gene cluster, there are 1 to 10 known, depending on which different organisms, there are different Cas9s. And these Cas9 genes express Cas proteins that actually help bacteria in fighting viruses and phages. And the third component that we have is tracer RNA, which actually helps in processing of CRISPR RNA that is produced from CRISPR locus. So these are the three component system. So how, let's just go to briefly see where and when they were identified and described. So the first time the CRISPR locus was detected in E. coli by Ishino's group in 1987. However, he didn't know what these sequences are and what were they doing in bacteria. So then in 1993, Francisco Mojica actually discovered, he analyzed short DNA fragments and found that these repeats are present even in archaea. Then he got fascinated with this and he wanted to find out what these are. So he actually started working and he started analyzing different molecular microbes and found in most of, like in about 20 different microbes, including mycobacterium and plague bacteria, he found that these CRISPR loci was present. However, the sequence were random and different. They were not conserved. Then while he was working on finding these sequences, another person, Jensen, he discovered that there is a CRISPR cluster which is just before the CRISPR loci. And he found there were 4 genes, Cas1 to Cas1, 2, 3, and 4, Cas protein gene. However, he couldn't uncover the function of these genes. Then again in 2005, Mojica went out and Bolotin's group discovered independently that the CRISPR locus actually contain part of the random spaces that are there. These random spaces are actually part of genetic material of phages that or viruses that infect bacteria. Then in 2006, for the first time, Markov showed that there is a protein called Cas9, which is actually actively involved in bacterial immune system. However, he couldn't pinpoint the molecular function of this protein. So finally, in 2007, Barangao actually experimentally showed that the CRISPR-Cas9 system is an adaptive immune system in bacteria and archaea. So what he did, basically, he deleted the CRISPR locus and then found that these bacteria are actually susceptible for 2 bacteriophage infections. And finally, in 2011, Emmanuel Charpentier's lab discovered that there is, apart from this CRISPR RNA, there is another RNA which is called tracer RNA. That RNA is required and for processing of this CRISPR RNA. So if you don't have this tracer RNA, somehow the CRISPR RNA is not able to defend the infection from phages and viruses. And in 2012, the Carpentier's group and Jordan's group, they worked together. And independently, Sixnick's group also work on the similar thing. And then the Carpentier and Dodna for the first time showed that it's the tracer RNA, which it actually forms a complex with the CRISPR RNA and together stabilize, these two together stabilizes the Cas9 nuclease with them, which actually then creates a double-stranded break in DNA. So this was the first time, this was discovered for the first time by Charpentier and Dodna. However, since Sixnick also came up with a similar study, a similar result, but his paper came out about a month after Charpentier and Dodna's. And then in 2013, January, for the first time, Feng used the system to edit mammalian genome, basically. So this is all about CRISPR, discovery and all. So how does it work actually? So sorry. So yeah. So now we can see that a lot of more than 50 modern organisms actually use CRISPR and Cas9 system for gene editing, and you can see here that from 2012, and these 4 things came out, and now if you compare, there is a huge increase in publications that actually use this system for various different model system, and they have developed many different other techniques based on CRISPR-Cas9. And actually the paper Charpentier's and Jennifer's paper was actually is cited almost about 4,000 times since then. So it's all about now CRISPR. How does it work? What is CRISPR-Cas9 and how does it work? So CRISPR-Cas9, as I told you, that it's an adaptive immune response from bacteria towards bacteriophage. So what happens? So in a prokaryotic cell, when there is a phage infection, what happens? There are two proteins that are expressed, Cas1 and Cas9. They will go and detect this foreign DNA, and they will find and look for one specific motif, which is called protospacer agitator motif. It is NGG. So Cas1 and Cas9, they recognize this motif, and then they bind to this foreign DNA, and then they start chopping this DNA. And then they grab small pieces of this foreign DNA, and then they insert it in CRISPR locus between the two palindromic repeats. So these palindromic repeats, here what we show that they are actually, these pieces are from different bacteria or different bacteriophages and viruses. Now, once it is done, if there is another attack from this bacteriophage, what happens? CRISPR locus is transcribed, where you have a long RNA, but as I said, this repeat is palindromic. These will form a hairpin loop. So now we have one hairpin loop and one spacer, another hairpin and another spacer. So this is how a pre-CRISPR RNA is transcribed. And then other components of Cas9 1 to 6, depending on which bacteria it will process this CRISPR RNA into small pre-CRISPR RNA into small CRISPR RNA. Now this small CRISPR RNA is being made, and at another locus where we have tracer RNA locus, tracer RNA will be transcribed, which will travel, and then another Cas9 protein is expressed. So this is an asparagine. That's what, depending on other bacteria, there are different complexes of Cas9s. So Cas9 or other 6, 8, or 10 Cas big complex, it is expressed. That together with, so CRISPR RNA together with the tracer RNA forms a complex, a stable complex, so that Cas9 then recognizes and binds to it, and then they go and bind to target sequence based on the sequence similarity we have here, and then CRISPR is activated and it creates a double-stranded break and degrades this phage DNA. Okay, so this is the system. So here what Jennifer and Emmanuel's lab finally found is that tracer RNA is important for this complex formation, and Cas9 actually depends on this complex to get stabilized on the DNA, phase DNA, and create a double-stranded break. Right. So this is how the CRISPR system works in the basically bacteria. So then when they were doing this, they thought, how can we use this system outside of bacteria to perform genome editing? So what they did, they started looking, they started doing in vitro experiments where what they did is they expressed, they brought Cas9 and then a CRISPR RNA, which has complementary sequence to a target RNA that they wanted to degrade or cut, and then a tracer RNA, which has nice binding to the CRISPR RNA. So when they bring these three components together, Cas9 was able to degrade or cleave, make a double-stranded break in DNA. So this was a 3-component system. Then they modified it and brought 2-component system, where what they did is Cas9 is same. They fused these two RNAs, CRISPR RNA and tracer, and they created one single CRISPR RNA and tracer RNA chimera, which we call guide RNA. So then they created this two-component system and performed in vitro experiment and showed that it works. So I'll just show you one piece of data from this paper, where what they did here is they took a plasmid. There is they have the GFP here, and they created five different probes or guide RNA, chimeric guide RNA with close to PAM site. And they perform an experiment where they selected one restriction enzyme which will digest upstream of this site, and then guide RNA. They perform in vitro reaction together with Cas9, and they found that each and every reaction actually gave rise to this small fragment that they expect, like digest with Sal1, and there was another cut with the Cas9, and they could do it in vitro. So this gave them confidence that this can be used, the system can be used in many different model organisms. And today, what we do is we use the same strategy where we have the two-component system that was developed by them. In any model organism, what we do is we designed a guide RNA, which is complementary to the target RNA wherever we want to create a mutation. So we have a PAM sequence, and then close to PAM, we will select our guide RNA sequence. And then this is one component where, and then the Cas9 is another component. And then we express both of these things in different model organisms, and then it will create a double-stranded break. So what happens after double-stranded break? Once the double-stranded break is made, the cells can repair it into two ways. First is non-homologous end joining, where it will create small indel mutations, deletion, or a small or little large deletion. And then, or the other way is that we provide a template, DNA template, where we can give homology sequences and then get whatever any DNA that we want to get inserted in this. So this way we can basically tag any gene or create small or large deletions or totally change the gene sequence. Okay, so this is how it works in genome disruption way or gene disruption way. But now Cas9 CRISPR system is being used for many other things, not just only for creating double-stranded break like what we saw here. It is also being used for controlling or for checking gene expression or controlling gene expression. What they did is basically what here you can do is fuse a Cas9 enzyme with or transcription regulator, and you can decide on you can decide where you want to bring this transactivator. You will decide the guide RNA according to that, and then which can actually affect the expression of this gene. Another thing which is developed based on the CRISPR-Cas9 system is epigenetic modification, which was not possible before. So again, here with the help of guide RNA, you can stabilize this complex here. And in this case, Cas9 is fused to an epigenetic modifier, a domain that is involved in epigenetic modification. That's how you can actually bring these changes close to the target site. Then we can also look at RNA, monitor RNA. And for that, we will basically, so in these two cases, what we do is we do not, we use Cas9, which is nuclease dead, because here we want to create a double-stranded break, but here in this case, we do not want to create a double-stranded break. So we just utilize the part or the property that is actually binding to this complex. Then we have RNA targeting. Again, in the same way, we have nuclease dead, either Cas9 or there is another Cas protein which is being used is Cas13. So what you do is basically you target RNA, and again, either the Cas9 will have a fluorescent probe that you can monitor, or somehow these small RNAs are modified. So they carry certain motifs that can bind to different fluorophores, and this is how you can modify or you can monitor RNA trafficking. Then we have a new method that was again developed on CRISPR-Cas9, which is to change the chromatin topology. So chromatin topology changing, normally you cannot do it. So far, I didn't see any system that does it very like this precisely. So what here they did is that two proteins, they interact with each other and they will bind to different sites. So this way you can bring promoter and enhancer. So basically bring two components that are far away from each other, you bring them together so that now promoter and enhancer, they can interact with each other, and then you can look at the gene expression profiling. Okay, so this was all about all these methods that are CRISPR-Cas dependent, and they are involved in basic, we use them in basic understanding basic biology. But apart from basic biology, we actually have more for CRISPR-Cas9. So people have used CRISPR system for crop improvement. More than 20 crops have been, in more than 20 crops, people have used CRISPR-Cas9 system to modify these to basically fight with viruses and mold infection, or to basically fight with drought or salinity problems. So far, these are all experimental in laboratory. However, there are three cases like the maize, where is the vex corn, and for soybean and butter mushrooms. These three have been approved, so that there are changes. And then they have been approved by, at least in USA, they are at the large production. They will come out in market soon. And apart from this, we also have CRISPR in medicine. So in diagnostics, there are kits that were based on CRISPR-Cas9 interaction. So there are kits for Zika virus and MRSA, dengue virus, and African swine flu virus, and also COVID. And one of the COVID kit is actually developed in one of the CSIR laboratories, which is IGIB in Delhi. So apart from diagnostics, people are trying to use CRISPR-Cas9 in curing diseases also. There are clinical trials going on for sickle cell anemia, beta thalassemia, and a rare eye disease, which is LCA10, and also for cancers. So we can see already that, CRISPR is involved in not only in the basic biology, or important not only in basic biology, but also in crops and medicine. So overall, CRISPR system has taken our life sciences into a new epoch. And it is actually in many ways bringing great benefit to humankind. So I would say that it is one of the best discoveries in the this field, genome editing field so far. And I am sure that we almost everybody is using this system. All right. With this, I would like to finish. Thank you.
Thank you, Sonal, for this informative talk, giving us an overview of the CRISPR-Cas9 system. We have a couple of questions. There's a question which we have got from messages. The gene editing using CRISPR technique seems like a magic wand. Is there a way forward to provide solutions to cure genetic diseases? And what is the way forward for human gene editing that is in line with laws drawn by scientists and governments?
So actually, people are trying human gene editing. There are basically 4 different studies where actually people tried for in human embryos for changing, genetic, for changing the different genes. So far, it was not, it's not great, because CRISPR-Cas9 also bring one problem, that is off-target effects. So there were when these people looked at embryos after gene editing, they found there were mutations and there were arrangements in many different other loci. So for that, they are developing, like there are people, like scientists, who are developing modified versions of Cas proteins that actually have less off targets. So it's going on for, as I said, clinical trials for 4 different studies are, like 4 different diseases are going on. So we'll see in future.
Okay. Is CRISPR going to push us in a fast-paced evolution mode? This is another question which has come through messages.
Can you repeat it? Sorry.
Is CRISPR going to push us in a fast-paced evolution mode?
Fast-paced evolution mode. Well, it depends on us. If we control things, then no.
There's also another question which we have just received on YouTube. Are there any limitations for this technique? Can it be done on fetuses?
So fetuses, okay, there is one study or one case that was reported by a Chinese group scientists. They actually tried CRISPR experiment on human embryos, and then actually the two kids are already born. So what they did is they deleted one gene that actually lets HIV virus enter cell. And but so far we do not know, because they didn't look at what were the side effects of these. So I don't think we are going to do it in near future. As I said, there are other three studies where they found large deletions and large rearrangements. So I don't think it is going to be very soon that we can start experimenting on humans,
There's
especially
another
fetus.
question. Sorry. Yeah. So
Especially
there's,
fetus, because it changes everything. Localized effect is one thing, but here, the whole organism is developing from that fetus. So it will change a lot of different things.
there's another question. How is the guide RNA created?
So guide RNA, so what we do basically, we like normal PCR, you get the sequence, you analyze the sequence, you find the locus where you want to make change, and then there is the DNA sequence. You actually get complementary of that sequence. You can order, you can get it synthesized from companies. So there are ways you can simply get those.
Thank you so much, Sonal, for this talk and answering so many questions which had come through both YouTube and messages. If there are any other questions which we receive later, we would be happy to send it to you and then maybe share it with whoever has asked the questions.
Sure.
And thank you so much for your time.
Thank you.
And now we will move on to some other questions which are still left. May I request Ajith to take some questions which we have received for his talk on black holes?
Sure.
So, have astronomers made any observations which do not agree with the general theory of relativity?
It's an interesting question. There were some puzzling observations. One was an observation made by the Pioneer satellite. But it turns out that this could be explained by an experimental error. So far, all such observations that suggested deviations or violations of general relativity could be tracked to experimental or observation errors. So far, there is no conclusive evidence that require any modification of GR. There is this puzzling observation that the universe is expanding at a speed, at a pace that is faster than that is predicted by our known matter content in the universe. But the most popular explanation of that is that there is some unknown form of energy called vacuum energy or dark energy that we do not know yet. Some people also try to explain this by arguing that the GR, general relativity, gets modified at very large scales of cosmology. But I would say there is no conclusive evidence that suggests that GR needs to be modified.
Okay. So there's another question. Can these supermassive black holes act as seeds for galaxy formation?
It seems that supermassive black holes seems to play a major role in the formation and evolution of galaxies. But are you suggesting that the stars assemble themselves around supermassive black holes? That is unlikely, because if you want to produce black holes, you want to start with some stars. So I don't think supermassive black holes are playing a role of seeding galaxies, but they seem to be high. So, for example, there is this very interesting observed relation that the mass of the galaxies is highly correlated with the mass of the central supermassive black hole. So there is some empirical evidence that suggests that supermassive black holes are playing a major role in the evolution of galaxies. But the details are not very clear, and admittedly, I'm not an expert in this topic as well.
There's another question about what is the lifespan of a black hole? Will they live on forever or they end up transforming into something else?
Yeah, so according to classical theory of physics, classical theory of gravity, black holes will live forever. But if you take into account some quantum mechanical calculations, which are very hard to do because you don't have a fully quantum theory of gravity yet, but one person or a few people, notably Hawking and Beck-Einstein, tried to do a semi-classical calculation, which is bringing some aspects of quantum mechanics to the calculation of black holes. They found that black holes can actually radiate some energy outside, which is against the popular notion of black holes. And in that case, black holes can actually lose energy and kind of disappear. But it turns out that the amount of Hawking radiation is inversely proportional to the mass, the large power of the mass of black holes. So for the black holes that we actually observe in nature, the astrophysical black holes, the Hawking radiation is completely negligible. But if there are some extremely tiny black holes, like the kind of primordial black hole that could have formed in the early universe, some of these very light primordial black holes could have completely disappeared by evaporating into Hawking, by emitting Hawking radiation.
So there's another question. What is the next big thing in observational astronomy after gravitational waves and James Webb Telescope?
That's a very hard thing to say, and it's probably a matter of taste and interest and exposure. There are lots of interesting things coming up. One is that the horizon of astronomy is increasing significantly. It started with optical light. It went through the full electromagnetic spectrum, neutrinos and cosmic rays started coming, now gravitational waves. So the people are starting to use different messengers to probe the cosmos, and they all probe very different aspects of the astrophysical phenomena. And each of these messengers, people are pushing the frontiers, both in neutrinos, astrophysical neutrinos, gravitational waves, X-rays, or cosmic microwave background observations. Transient astronomy is another big field that is using optical telescopes. But earlier, people were used to observing the sky as a static object. But we know that there are a lot of things that are changing. There are transient phenomena in astronomy, in the cosmos. There is a huge amount of attention going to the study of this transient phenomena, like exploding stars, GRBs, and so on. There are large radio telescopes coming up, like the Square Kilometer Array, which would probe a number of interesting things. One is probing the so-called dark ages, and there is, for example, some observational gap in the probes of the universe. Before the formation of first stars and after the observation of what is called the cosmic microwave background radiation. So there are proposed X-ray telescopes. There are improvements on the neutrino telescopes like IceCube proposed. So there is a variety of interesting things happening in astronomy, observational astronomy. So you can pick your favorite.
Next question. Einstein's equations can predict black holes, but why can they not predict what happens after that?
What happens after that? You mean after is some reference to time, or you mean what happens at the interior, the singularity of the black holes?
I'm guessing it's the latter, but I can confirm from the
right. It turns out that if you're talking about a singularity, it turns out that these Einstein's equations actually encounter a singularity, which means that you cannot solve them at that point. So a lot of people believe that there has to be some substructure, some kind of a quantum phenomena that would resolve that singularity. And since Einstein's equations are coming from a classical theory of gravity, they have no notion of quantum mechanics, no quantum physics. So you have to have a quantum theory of gravity to describe what is happening in this very small region. And there are many candidate theories, but all of them are in the works.
There's one last question. How does the ground-based telescopes using adaptive optics compares to the space telescope, where atmospheric disturbance is absent as a whole?
That's a very good question. So, even if you use adaptive optics, the best that you can get is to the quality of space telescopes, because the space telescopes don't have to deal with atmosphere. But where the ground-based telescopes have an advantage over space telescopes is because of their larger collecting area. You can build a much larger telescope the same price as a space telescope, and it's very hard. It's much more expensive to launch a very large space telescope to space. For example, Hubble has a diameter of 1 meter or 2, while even the current telescopes like Keck and the VLT, very large telescopes, has diameters of 10 meters or so. And there are plans of building 30-meter telescopes and 40-meter telescopes, etc., because these are much more cost effective. So the summary is that the ground-based telescopes have the advantage of the larger collecting area, while the space-based telescopes have the advantage of no atmosphere.
Okay, so I think we don't have any other questions about from any other topics yet. So I think we'll bring this program to a close now. I'd like to thank all the speakers for their time and for giving such wonderful talks today. And also a big thank you to the audience for attending this session. We hope to see you in the next Sawaal-Jawaab, that is on November 21st, where Ullas Kolthur is going to talk about when, what, and how much to eat. So once again, I thank the speakers and the audience for the speakers for giving the talks and the audience for attending and participating in this program. Thank you so much.
Thank you. Thank you for listening, and thank you for organizing.